Hello, my name is Maya Spivey and I am in Group 6. A microbiome is the total number of all genes encoded by all microbial genomes. The gut microbiome consists of microorganisms located inside the gut such as bacteria, bacteriophage, and others. A physiological condition that has a relationship with the gut microbiome is Crohn's disease. Crohn's disease is an inflammatory disease that affects the lining of the digestive tract. Scientific studies have examined the roles of the microbiome with patients that have Crohn's disease. Researchers have studied the relationship between the two and stated that a bacterial enzyme causes there to be an imbalance in the gut microbiome. This research studied the relationship and focused on the relationship of the gut microbes and the amino acids in patients who have Crohn's disease. The results were there were high levels of amino acids present in stool samples from people who have Crohn's disease, and these results were also correlated to high levels of proteobacteria, which is a gut bacteria. This relationship is correlative. Another physiological condition that is studied with the gut microbiome is obesity. Obesity involves excessive body fat, which can lead to health risks. Studies have shown that obesity is linked to the composition and the function of the human microbiome. The human gastrointestinal tract has a large number of gut microbiota, including bacteria, fungi, protozoa, archaea, and many others. An experiment was conducted with germ-free mice, and these germ-free mice were raised in sterile environments, and they had no microorganisms within their gut. The digestible gut was taken from regular mice and transplanted to germ-free mice, and there was a 60% increase in body fat within 2 weeks without a diet change. These results concluded that the microbiota has impact on obesity. This relationship is causal. These two outside sources are for Crohn's disease, and this source is for the obesity. Our hypothesis was where the gut microbiome results from a rabbit be different from the gut microbiome results from a ferret due to differences in families and diet. The question for our experiment was, will the DNA results between a rabbit and a ferret be different due to differences in families and diet? For our materials and methods, the animals that we decided to compare were 2 rabbits and a ferret. The 2 rabbits were ironhead rabbit, New Zealand rabbit, and the ferret that we compared was a cinnamon ferret. There are some differences and similarities between the ferret and the rabbits. Similarities include like size; they're both very small animals with fur. But there are more differences than similarities. So differences includes families, which is the main difference. The families of the rabbits are the leproids, and the family of the ferrets were mustelids. Also in diet, ferrets eat meat with vegetation, and rabbits are herbivores. For this experiment, we compared diet and family, and we first isolated DNA with the Keegan kit, and then we used the polymerase chain reaction to assess how much DNA came. So we, to briefly summarize the protocol, we just took 3 poop samples. We thoroughly, we thawed them out, and then we weighed them, placed them in microcentrifuge tubes, and then we added buffer and water to it, and then we placed them in the centrifuge and allowed it to go at its highest speed for 1 minute. And then we added 500 microliters of the proteinase K and the supernatant from the centrifuge samples. And then after adding the buffer, we vortexed it for 10 seconds until it was thoroughly homogenized. And then we added 750 microliters of lysate to the Keegan spin column, and we discarded the filtrate after we added 500 microliters of wash buffer to the waste beaker. After adding 500 microliters of another wash buffer, we then added it to the centrifuge and allowed it to work for 3 minutes at highest speed. We then added 1.5 microliters of the Keegan spin column to a new tube and added 200 microliters of ATE. The amount of DNA we received for the samples were 20.35, 12.8, 14.54, 14.16, 20.21, and 17.83. For the samples that are closer to 10, they would assume to have more DNA, and for the samples that are closer to 20, they have a smaller amount of DNA in them. We used a qPCR, and it was not handled by us. It was by our team. Like, we did not set it up ourselves. We put our our sample on it, and this cyber green was used to stain agarose gels to visualize the DNA that is present. We did follow this protocol. We didn't make any changes to it, and we did follow the instructions that were posted on Blackboard. So for our results, DNA absorbs light well at 260 nanometers and protein absorbs light well at 280 nanometers. And we can use the ratio of absorbance at 260 to 280 to measure purity of DNA. And these were the concentrations that we got. For the New Zealand white rabbit, we got 21.9. For lionhead rabbit, we got a concentration of 32.3. And for the ferret, we got a concentration of 14.5. This right here is the amplification curve from class. This green, or this thick line right here, that is the threshold in which the, so for the threshold, these lines need to cross it for the data to be usable in the QC or QT values. And the cycle numbers that are closer to 0 will have a higher abundance of the targeted DNA than the numbers that are farthest away from 0, which could contain contamination. These are our 3 graphs. This graph is eubacteria, the eubacteria template. This one is the ratio of eubacteria firmicutes, and this is the bacteroidetes and eubacteria ratio. For our discussion, when there's a high enough concentration of DNA, that means that pure DNA has a ratio of 260 to 80 of approximately 1.5 to 2.2. Ratios below or above this range could indicate possible contamination of proteins. If the ratio is far from the acceptable range, then the estimate of the DNA concentration will most likely be inaccurate. The range of the ratio for our samples were 2.17 and 2.53, with the rabbit 2 being the lowest and rabbit 1 being the highest. For the template that we only tested eubacteria, our cycle numbers were adequate for our analysis. All the samples were higher than our negative control and lower than our positive control. For the second template, when we did the ratio between the eubacteria and the firmicutes, the negative control is good and the positive control is great for the primers that we use. And all of our samples are greater than the positive control because we were given the wrong primers that matched with E. coli, which was the positive control. And E. coli is not part of the phyla for firmicutes and bacteroidetes. For the last template with the ratio of bacteroidetes and eubacteria, something was wrong. Either our calculations are off or we did not remember correctly which sample was in which when we put it through the qPCR. Rabbit 2 was extremely high and rabbit 1 and ferret, and the ferret results were 0.01 off from each other, which has never happened in the previous templates. The positive control is really low and the negative control is greater than the 2 samples, thus making the numbers inadequate for our analysis. Comparing the first 2 graphs, the ferrets had less firmicutes and bacteroidetes than both of the rabbits. So this is the ferret and these are the rabbits. This is the ferret rabbits. This is the ferret rabbit. So you can see that the rabbits are higher than the ferret. In the future, we would like to test other, since it is the eubacteria, the ferret has to have some type of bacteria more than the rabbits. It is just a different phyla than what we test in this experiment. In the future, we would like to test other phyla, so maybe we can determine what kind of bacteria ferrets have more than rabbits. So for the differences between the gut microbiomes of the animals that we compared, like I said, the eubacteria was higher for the rabbits, and then also the firmicutes were slightly higher, and also for the physiological disease I was talking about, or conditions I was talking about. Crohn's disease has high levels of proteobacteria, and for obesity it had high levels of bacteroidetes. So for our results, our data Our data did answer our experimental question and support our hypothesis because it showed that there are differences within the bacteria due to differences of the families of the rabbits and the ferrets. Rabbits were in closer range with one another, but ferret were not. In future experiments, we could also choose another experimental question and hypothesis so we can get the most out of our experiment and truly understand what we are looking for and comparing. Also to write down exactly what the actual numbers are for the qPCR template in which we place our samples in. Here are our pages. Our graphs. Thank you so much for your time.